Welcome to our deep dive into the fascinating world of aging. Today we'll explore what aging is, why it's crucial to study it, and delve into various theories and mechanisms that drive the aging process. We'll also discuss cutting-edge research and potential interventions that may one day help us slow down or even reverse the effects of aging. Let's get started. Aging is a physiological and dynamic process that begins in the fourth decade of life and continues until death. It affects us biologically, psychologically, and socially. As time progresses, our bodies undergo a series of changes, leading to the gradual decline of physical and cognitive functions. This process is complex and individualized, meaning everyone ages differently based on their genetic makeup, lifestyle, and environmental factors. So why should we study aging? The answer lies in the significant impact aging has on health, society, and the economy. Old age is the greatest risk factor for chronic diseases responsible for most morbidity, mortality, and healthcare costs worldwide. These diseases include atherosclerosis, cardiovascular diseases, strokes, dementias, cancers, neurodegenerative diseases, diabetes, and others. According to recent estimates, a mere 2% delay in the progression of aging processes, compared to doing nothing or curing a single disease like cancer or heart disease, would result in 10 million more healthy, rather than disabled, elderly people in the United States alone. This delay could save an astounding $7.1 trillion over the next 40 years. These numbers highlight the tremendous potential health and economic benefits of targeting the fundamental mechanisms of aging, rather than treating each disease separately. Aging is caused by multiple interacting processes. Here are some prominent theories about aging. Programmed theories suggest our bodies are programmed to age. They include gene theory, endocrine theory, and immunological theory. Error theories propose aging results from accumulated cellular damage, including wear and tear theory, free radical theory, and mitochondrial theory. Genetic theories focus on the role of genes and states that our lifespan and the aging process are largely determined by our genetic makeup, such as telomere theory and stem cell theory. Evolutionary theories like mutation accumulation and antagonistic pleiotropy suggest aging is a byproduct of evolutionary processes. Biochemical theories propose that biochemical reactions, like glycation and heat shock response, contribute to aging. The geroscience is an interdisciplinary field that aims to understand the relationship between aging and age-related diseases and disabilities. It takes a different approach, seeking to understand the genetic, molecular, and cellular mechanisms that make aging a major risk factor and driver of common chronic conditions and diseases in older people. This approach aims to therapeutically target these fundamental aging mechanisms to impact multiple chronic diseases simultaneously. One fundamental aging mechanism that underlies numerous age-related diseases is cellular senescence. Senescence is a state where cells lose their ability to divide and function properly, becoming resistant to cell death and exhibiting increased metabolic activity. A permanent arrest is effective to ensure that damaged or transformed cells do not perpetuate their genomes. This growth arrest is implemented by the activation of p16 and p53 tumor suppressor pathways. Senescent cells adopt a pro-inflammatory phenotype, secreting factors that can damage surrounding tissues and promote chronic inflammation, known as the senescence-associated secretory phenotype called SAS. Cellular senescence contributes to aging by promoting tissue dysfunction and chronic inflammation. The removal of these cells has been shown to improve health span and even extend lifespan in animal models. This has led to the development of senolytic therapies, which aim to selectively eliminate senescent cells to mitigate their harmful effects. Let's delve into the primary causes of cellular senescence. Epigenetic factors. Epigenetic is the study of changes in gene expression that don't involve alterations to the DNA sequence. Essentially, it's about how genes can be turned on or off by chemical modifications such as DNA methylation and histone modification. These changes can accumulate with age, leading to altered cellular functions and contributing to the aging process. Research has shown that certain patterns of DNA methylation are associated with age and can even be used to predict biological age, sometimes referred to as the epigenetic clock. This clock can provide insights into how fast or slow someone is aging at a molecular level. Further, it is found that DNA methylation is generally downregulated in a variety of mouse tissues and human fibroblasts during aging. Telomere erosion. Telomeres are repetitive DNA sequences at the ends of chromosomes that protect them from damage. With each cell division, telomeres shorten until they reach a critical length, termed the Hayflick limit, which triggers the DNA damage response (DDR) and activation of the tumor suppressor p53 pathway, resulting in either apoptosis or exit from the cycle. Interestingly, a study published in 2024 on astronauts have shown that spaceflight can cause rapid telomere shortening upon return to Earth, potentially accelerating aging and increasing disease risk. DNA damage. DNA damage is a significant trigger of cellular senescence. The DNA damage response (DDR) activates tumor suppressor pathways, leading to cell cycle arrest and senescence. Persistent DNA damage can result from various factors, including oxidative stress and environmental toxins. Mitochondrial dysfunction. Mitochondria are the powerhouses of the cell, producing energy. However, as we age, mitochondrial function declines, leading to increased production of reactive oxygen species rose and cellular damage. This dysfunction can induce senescence and contribute to the aging process. Researchers and pharmaceutical companies are exploring several pathways to develop gerotherapeutics that target the mechanisms of aging. Targeting impaired autophagy. Autophagy is the process by which cells recycle damaged components. Inhibiting the mTOR pathway, for example with dietary restriction or rapamycin, can enhance autophagy and extend lifespan in various organisms. The randomized, double-blind, placebo-controlled trial PEARL was recently initiated in the US to assess the long-term efficacy of rapamycin in reducing clinical outcomes associated with declining health and aging in healthy old adults. And cellular senescence. Senolytic drugs aim to eliminate senescent cells, potentially improving health span and lifespan. It is important to mention here that aspirin was once thought to have potential as a xenomorphic drug, that is, the drugs that suppress the effects of SASP components secreted by senescent stem cells. However, recent data from the ASPRI large clinical trial found that daily low-dose aspirin in healthy older adults without previous cardiovascular events does not prolong healthy and independent living, but surprisingly might predispose to higher mortality by cancer. Based on this finding, the guidelines of the American Heart Association recommend against the routine use of low-dose aspirin in this category of old adults. Nuclear RNA polymerase II. A recent study identified nuclear RNA polymerase II as a therapeutic target, starting from the interesting observation that the transcriptional elongation speed of the enzyme is increased with aging across multiple species, including Drosophila melanogaster mice and rats and certain human cell lines. As a consequence, transcriptional errors, modifications in splicing, and the appearance of inefficient RNA forms might occur and impact the function and lifespan of affected cells. Therefore, slowing down its elongation speed through dietary restriction or genetic modification has been shown to extend lifespan in model organisms. In 2023, Pfizer Ventures joined VitaDAO to fund longevity research projects and foster the spin-outs of companies conducting research in the area of longevity. This collaboration highlights the growing interest in geroscience and the development of anti-aging therapies. Can we slow the effects of aging? Though aging is inevitable, it's possible to slow down some of its effects through healthy lifestyle habits. Here are some tips to help you age gracefully. Eat a nutritious diet, stay active, avoid tobacco and alcohol, attend regular checkups, know your family history, and engage your brain. Do our thoughts affect aging? There was a study done by Harvard scientists where they took a group of people in their 70s and 80s. The researcher asked these men for 5 days to pretend that they were 22 years old, thinking that way, acting that way, and feeling that way. She put the pictures of Kennedy brothers, Marilyn Monroe, and magazines that reminded them of being 22 years younger. She took multiple measurements at the beginning and end of the 5 days and found that their finger and toe lengths were longer, their arthritis was better, and their cognitive abilities improved by 60%. So you are the person you pretend to be. Think positive and stay positive as your body responds to the power of your thoughts. Understanding the complex process of aging and the mechanisms behind it is crucial for developing interventions that can improve health span. By targeting the fundamental drivers of aging, we can hope to mitigate multiple chronic diseases simultaneously, leading to a healthier life. Thank you for joining us on this journey into the science of aging. Stay curious, stay healthy, and see you next time.